In this video, we're going to talk about keto-enol tautomerization. Ketones and aldehydes have two possible forms, a keto form and an enol form. The process for converting from one form to the other is known as tautomerization. If you have the keto form and you are going to the enol form, it's referred to as keto-to-enol tautomerization. And if you have the enol form and you're going to the keto form, it is referred to as Enol-to-keto tautomerization. The keto and the enol forms are related to the two resonance structures you see here, where the negative charge can be either on the oxygen or on the carbon. These are the two resonance structures of the enolate anion, and the one with the negative charge on the oxygen is more similar to the enol form, and the one with the negative charge on the carbon is more similar to the keto form. If you were to add arrows to our resonance structure here to get from this form where you have the negative charge on the oxygen, you would draw a bond showing that lone pair moving down and becoming the carbon-oxygen double bond. And then you would have another arrow taking this carbon-carbon double bond and having it become a lone pair on that carbon. So if you were told, hey, I have this molecule and I have to draw its resonance structure, you would show that lone pair moving down to form the double bond, and you would show the carbon-carbon pi bond moving over on the carbon to form a lone pair. And also, if we were going to talk about which of these would be favored, remember we did that in chapter 6, the negative charge would prefer to be on the more electronegative atom. So this resonance structure with the negative charge on the oxygen would be the major resonance contributor, and this one down here with the negative charge on the carbon would contribute the least. You can see here that the keto form is typically favored and generally dominates the equilibrium. If you're looking at the percentages here, we can see that the keto form for this example down here exists in almost 100%. The same thing is true for this bottom one over here. Again, the keto form exists almost 100%. So usually if we see a ketone, that is the preferred form. Now keep in mind, a given ketone may have one or more different possible enols. The aldehyde examples shown here only have one possible enol, but you can imagine that if you had a ketone like this, you would have two potential enol forms, because you could have the double bond on the left or on the right. So the first version here would be the keto form, and then this would be enol form number 1 and enol form number 2. Okay, so let's look at the mechanism for keto-to-enol tautomerization, or keto-to-enol formation. We're going to see this happen under two different types of reaction conditions. This first one we're going to look at is basic conditions. That means that we're going to have a base, in this case hydroxide, involved in the keto-to-enol tautomerization. The first thing that happens is the base deprotonates this hydrogen here. We refer to that hydrogen as the alpha hydrogen because it is attached to the carbon next to the ketone. So I'm going to color in the electrons here. Our red lone pair on the hydroxide becomes an oxygen-hydrogen bond. The carbon-hydrogen bond here becomes a blue lone pair. So that's our first step, forming an oxygen-hydrogen bond to remove that proton and moving the carbon-hydrogen bond onto the carbon to form a lone pair. Next, you can see that we're just doing resonance. These are resonance structures where we're pushing that lone pair to form a carbon-carbon double bond. Then the carbon-oxygen double bond becomes a lone pair on the oxygen. So the blue lone pair becomes the carbon-carbon double bond, and the carbon-oxygen bond becomes a lone pair on the oxygen. The last step is a proton transfer step. So now that O- needs a proton to become the enol form. And that green lone pair is going to come over here and pick up a hydrogen from the water we formed in step 1. That's going to cause the hydrogen-oxygen bond to break and put a red lone pair on hydroxide. So if we follow the electrons from that last step, we use that lone pair on the oxygen to form a hydrogen-oxygen bond, and that caused the hydrogen-oxygen bond on water to break and become a lone pair on hydroxide. Notice that we started off with hydroxide and reformed hydroxide, so it is considered to be a catalyst in this reaction. We can also go from the keto form to the enol form under acidic conditions. In acidic conditions, instead of a base, which has a negative charge, we're using an acid, which has a positive charge. So all we're going to do is flip the steps. In basic conditions, we deprotonated first and then protonated after our resonance step. Here we're going to protonate first and then deprotonate after our resonance step. The key thing with basic conditions versus acidic conditions is to remember that in basic conditions, we're going to have negative charges, no positive charges, and in acidic conditions, we have positive charges and no negative charges. So that's how we keep track of what's going on. Okay, let's get this started here. The lone pair on the ketone becomes the oxygen-hydrogen bond over here on the right. The oxygen-hydrogen bond in hydronium, H3O+, becomes a lone pair on water. Okay, so this lone pair here becomes a bond, and this bond becomes a lone pair. This is a proton transfer step. We're just protonating that ketone. Now all we're doing in this next step is resonance. We're taking that oxygen-carbon bond and pushing it up onto the oxygen. When we do this step, the oxygen is gaining electrons, so it loses its charge. And then the carbon lost electrons, so it now has a charge. Now that we've done the resonance, we need to do our proton transfer step where we deprotonate that alpha hydrogen. The water is going to come over here and pick up that hydrogen. It's not drawn out here, so I'll draw it for you over here on the right. So water is picking up that hydrogen and forming a new oxygen-hydrogen bond. And that's going to cause that carbon-hydrogen bond to break and form a carbon-carbon double bond. The hydrogen fell off and became attached to the water to form hydronium, H3O+, and the carbon-hydrogen bond broke to become a double bond on the enol. Now let's look at the mechanism for Enol-to-keto tautomerization, or Enol-to-keto formation. We're just going to do the reverse of what we did before. In the previous example, we did our deprotonation of the alpha hydrogen, then our resonance, and then protonated the oxygen. And we're just going to do the reverse. So we're going to deprotonate the oxygen, then do the resonance, and then protonate the alpha hydrogen. Let's follow the electrons. So we have our red lone pair over here on hydroxide, forming a new oxygen-hydrogen bond. The hydrogen-oxygen bond comes down to form a blue lone pair. So that's our first step. Lone pair on hydroxide becomes an oxygen-hydrogen bond, and the hydrogen-oxygen bond on the enol comes down to the oxygen to form a blue lone pair. In the resonance, the blue lone pair comes down to form an oxygen-carbon pi bond. The carbon-carbon pi bond goes over to form a lone pair on the carbon. So we're bringing this pair of electrons down to form the oxygen-carbon double bond, and the pi electrons go onto the carbon to form the negative charge. In the last proton transfer, that green lone pair comes over here to pick up a hydrogen, and that hydrogen-oxygen bond breaks to put a lone pair on hydroxide. So we're just forming one bond here to the new hydrogen, and then the oxygen-hydrogen bond is breaking to put a lone pair on hydroxide. This can also happen under acidic conditions. So let's look at the acidic version of the reaction. Again, we're making sure that only positive charges are being used. These are all of our positive charges. The carbon-carbon double bond comes over here to form a carbon-hydrogen bond. Then the oxygen-hydrogen bond breaks to put a lone pair on water. So that's our first step, where those pi electrons become the carbon-hydrogen bond, and the oxygen-hydrogen bond becomes the lone pair. Our next step is resonance, where we have the lone pair on the oxygen coming down to form an oxygen-carbon pi bond. So that's our resonance step here. And then we're going to do our deprotonation or proton transfer step, where water comes in here, picks up a proton, and the hydrogen-oxygen bond breaks to put a lone pair on that keto form. Okay, so just to recap, water comes in to do that proton transfer. That blue lone pair becomes that oxygen-hydrogen bond there. Then the oxygen-hydrogen bond on our substrate breaks and becomes a pink lone pair over there on the keto form. Why is keto-enol tautomerization important? Well, this happens in your body all the time. For example, you can see this happening with sugars. Here we have the cyclic form of glucose, and it is phosphorylated because inside of cells, sugars are phosphorylated. Glucose 6-phosphate here can open up into its open chain form, undergo a tautomerization reaction. So here's the keto form, and here's the enol form. Undergo another tautomerization reaction, enol to keto, and this is a new keto form because we went from an aldehyde over here to a ketone over here, and then it opens again to form fructose. So this is how glucose can convert to fructose inside your body. Okay, this concludes Chapter 7. Please review the study guide for this lecture and let me know if you have any questions about any of the content we covered in this chapter.